Are you okay if we start?
Yeah, totally fine.
Okay, excellent. Good afternoon, everyone, and very welcome to the second webinar in our monthly series, Unravelling T-cell recognition, insights from immunology and AI. My name is Hashem Kui, and I'm delighted to be chairing today's session. This webinar series takes place on first Monday of each month, where we explore cutting-edge perspectives at the intersection of immunology and data science, including machine learning, AI, and systems immunology approaches. However, please note that our next session will be slightly shifted to Monday, 12th of May, as the first Monday of May is a bank holiday here in the UK. We are excited to be hosting Professor Wang Du Liu from Zhejiang University for our May webinar. And if you would like to be updated on future talks, please either follow me on LinkedIn, where I share announcement about this series, or please join the mailing list that I have created for this series. And if you find today's session interesting and valuable, I would really appreciate it if you could please help spread the word. The goal here is to foster an open, inclusive community for everyone interested in T-cell immunity, whether you are from academia, industry, or beyond. I'm also thrilled to share that we will be hosting an international conference and hackathon on this very theme at the end of September. It is shaping to be a very exciting event, and I would love to welcome you all on board. And you can see more details either in our website or follow us on LinkedIn again or other social media that we announce the news. And now it's my great pleasure to introduce today's speaker, Dr. Chris Thorpe. Chris is an EMBL Marie Curie Arise Fellow at the European Bioinformatics Institute and a visiting researcher here at MRC, Federal Institute of Molecular Medicine, University of Oxford. His research focuses on integrating data types with protein structure analysis and prediction to advance our understanding of T-cell immunity. Chris builds research tools and curated data sets for the immunology community, collaborating to co-design and test them in real-world applications. Originally a chemist, he holds a PhD in structural and computational immunology from Birkbeck College, University College London, and is a Fulbright scholar. Chris, thank you so much for joining us, and over to you.
That's lovely. Thank you, Hashem, and thank you everyone for joining us today. Hopefully by the end you'll know a lot more about MHC molecules and what data we have and where we might need some more if we're to really crack some of the problems that we're talking about in this series. I'm not going to spend too much time introducing the problem. I'm assuming that most people on the call sort of understand what we mean by T cells. But to give you the sort of like the quick overview that Alex gave so well last week, the bits that I'm really interested in are these MHC molecules, which are the ligands for T-cell receptors, and I'm particularly interested in the peptides that are bound to MHC molecules. And obviously in this diagram, we've got a CD8 T-cell, which is then going on to kill infected cells. But as I said, I'm expecting that most people sort of understand and know this sort of side of immunology, so I'm not going to cover it. What we're really interested in, and I think Alex gave a really great introduction last session, is this interaction between the T-cell receptor, which he likened to a barcode reader, with the peptide bound to the MHC molecule. And the barcode that the T-cell receptor reads is this combination of peptide and MHC molecule. And that sort of combination is going to be what my talk's about today. And we're all interested in this sort of 30-year-old grand challenge around antigen specificity, where we're interested as to why some T cells will see a peptide in the context of one MHC molecule, but the same peptide in another MHC molecule doesn't stimulate them. A completely different peptide maybe does, maybe doesn't stimulate them, and a slightly altered peptide stimulates them. It's a real puzzle and conundrum. And as I said, it's a 30-year-old grand challenge. If you're interested in just the MHC part of it, the first structure was published in 1987 by Don Wiley's group, with Pam Bjorkman doing a lot of the work there. And I recommend, go and read these historic papers, because there's always something interesting in them. The first complex of a human T-cell receptor with the peptide and again an MHC molecule was published in 1996. So it's something that everyone's been looking at for really a very long while. And if you're following the field currently, you'll have seen lots of these papers and preprints. We have a journal club in Oxford, along with Benji from Hashem's lab and Alex from Charlotte Dean's group and many others, where we look at lots of these. And we're moving forward, but the progress seems to be quite glacially slow at times. And it's such an important problem that Cancer Research UK has announced a grand challenge in 2023 to try and get some really sort of deep work into this problem. And Michael Birnbaum's team, the Matchmakers team, are sort of have been awarded the funds to look at this and are working hard. But there's room for the rest of us to look into it. And what we're really interested in is this really tiny interaction interface between this peptide bound to the MHC molecule that's seen by the T-cell receptor. So I'm going to break it down into talking about these barcode elements. And first of all, we're going to talk about MHC molecules. So there are two main classes of MHC molecules. I'm sure you're aware there's class 1 and class 2. So class 1 is found on all nucleated cells. It elicits cytotoxic T-cell responses, and it responds mainly to intracellular pathogens and tumours. And it's involved to some extent in autoimmunity. Whereas class 2 is found on these professional antigen-presenting cells. It's involved in helper or regulatory T cells, and it responds mainly to extracellular pathogens, but it can also see some tumor antigens through cross-presentation, and it's far more heavily involved in autoimmunity. And I'm going to concentrate today on class 1 molecules because that's where my main interest lies as a structural biologist. And I also curate a resource called Histo, which mainly looks at class 1 molecules at the moment, but the plan is to extend it to class 2 at some point. And so to give you a quick rundown of what Histo is, it's a fair data resource, so it's easily findable, accessible, interoperable, and reusable, all the content within it. And it's designed really to be a sort of a companion piece to SABDAB, which is a data source that Alex works on, comes from Charlotte Dean's group. And so that mainly focuses on the T-cell receptors, and I mainly focus on the MHC molecules. There are different ways to browse and search it. So you can browse by species, or you can use one of the searches. And when you get through to a structure page, then it's broken down to try and help you understand what information is within that structure. So there's obviously a very simple representation of it, which you can rotate. But there's also some analysis of the structure. So you can look at just the peptide. There are cutaways of the antigen binding cleft, so you can look at complementarity between the peptide and the antigen binding cleft. There's some information on where there are charges and hydrophobicity. So the hydrophobicity is in yellow, negative charges are in red, and positive charges are in blue. So you can get a really good picture of the sort of binding cleft that holds these peptides. You can also look at which residues in the MHC molecules are neighbors to particular positions within the peptide, so you can understand the interaction a bit more. And this is going to get a bit richer fairly soon by looking at hydrogen bonds between these, the different parts of the complex. You can understand the peptide binding pockets on the MHC molecule. As we'll see in a minute, all of the MHC molecules are highly allelic and polymorphic. And as a result, they form these binding pockets that bind the peptide. And then finally, there's some downloadable data. So this is about making it reusable. So you can download different parts of the complex, and you can use the sort of like the little clipped clipboard feature here to clip the URL for the structural components, and you can load, for instance, load them straight into something like PyMold, so you can do some molecular visualizations. You can also get all of the data that's behind the web page via an API as well, so you can again reuse it and use this in your analyses. And everything's organized into these collections of things called sets. So there are sets for a particular peptide sequence, and again you can get that as an API, and this gives you all of the structures that we know with that particular peptide sequence. You can also get a set of structures for a particular allele, and yet again you can get that data as an API. And this is just intended as a really quick overview, and hopefully you'll go and have a look and test it out and make suggestions to me. And there's a brand new version of the site and the pipeline that builds the data coming soon, which is a lot richer. So in the next couple of months, there'll be a very new version of the website that's got many of the analyses that sort of follow in this presentation. It's something I've been working on for quite a while, and we're nearly there for those of you who are interested. But you can use this structure this collection of structures to start doing some really interesting things. And a lot of what I'm showing is actually what you can do if you analyze bulk data. So we used to, when we look at a paper, we're seeing one structure compared to another, for example, or a few structures compared to another one. But actually, what can you do if you analyze bulk data? Well, the first thing we can do is we can see how amazingly biased our data is. We have an enormous amount of structures for HLA A0201, which is the predominant allele in white Western populations. And they're actually 40% of all the structures we have are of that one allele. There are some other alleles that we have a few more structures of, sort of compared to, the one or two we have for many alleles with diverse ancestry. But most of the structures we have are of sort of alleles that you find at sort of quite high copy number in white Western populations. And the first thing you can find when you analyze this data is that we need to do a lot better around diversity, particularly in terms of ancestries. Now, I've mentioned sort of alleles quite a lot so far. And so for those of you who are not really used to MHC molecules too much, the HLA, which is the MHC in humans, is both polymorphic and polygenic. So there are 3 class 1 loci, HLA-A, HLA-B, and HLA-C. And there are just over or around 3,000 alleles for each of these loci. And the polymorphisms are really surrounding this peptide binding cleft in the main. And so these purple regions are ones where you see polymorphisms. And they're also the positions that you see in the NetMHC binding predictor pseudo sequence that it generates. So there's a lot of diversity in MHC molecules in the human population. And this diversity gives us some really interesting things to do with health and disease. And one really lovely illustration of this is there was a paper in Nature back in 2023. So this is sort of looking at the pandemic, which is how lots of us sort of feel we don't have to explain what a T cell is quite so much. But what it showed was that there was this one human allele, HLA-B1501, which protects from severe COVID. And it does this by binding a peptide from a seasonal coronavirus that has a high sequence homology to the one from SARS-CoV-2. And so as a result of sort of conservation of this peptide sequence, if you've got B1501 and you've met this seasonal coronavirus before, you're protected from severe disease because you've already got a memory response there. And it happens in this particular allele and not others because of the residues around the P2 binding pocket, which hopefully you can see my cursor at the moment. And in B1501, there's quite a small amino acid at the entrance to the binding pocket, but in closely related alleles, there's a much larger tyrosine residue, and so that peptide can't bind. And this is just a really quick illustration about how these polymorphisms tune peptide binding preferences. and as a result of that, they tune the response to diseases.
Chris, I'm sorry to interrupt. We don't see your pointer or cursor if you are using it. Yeah.
I will try and work out what to do. Not sure how to change that. Do you see it now? Hashem?
No, I don't see anything yet, but it's okay. I think, I just wanted to let you know.
No, that's great. Thank you. Sorry about that, folks.
Not a problem.
Okay, so if we're talking about sort of peptide repertoires, then each of these alleles will bind a slightly different one. This is looking at 2 individuals from a flu trial that I'm involved in helping to look at the data for. And we can see that each of the different alleles, so, A2402, A3101, B3701, they all bind very, very different peptide motifs. And so as a result, there'll be a different repertoire of peptides that is shown to T cells, which is how you get a really diverse response within a population. And these motifs have been known about for quite a while now. But the interesting thing you can do with these motifs to find out how they cluster together and how different they can be is to look at their cosine similarities. So this is plotting the cosine similarities of all known motifs against all known motifs and looking at the data underlying them. And what comes out from this is there are some really quite striking blocks which are either similar or quite different. So up at the top left hand, we've got a really dark block which relates to HLA AO2. So this is the AO2 subtype, and we see that lots of the AO2 motifs are really similar. The next block is the sort of like the AO3, A11 block. Again, they're similar. But as we move into HLA-B, we find that the motifs are quite different to each other. And then when we look at HLA-C down the bottom right-hand corner, we can see that all the motifs are really quite disparate in terms of their similarity to each other and in their similarity to HLA-A and HLA-B. Looking a bit closer, just looking at the AO2 motifs, they're pretty similar in lots of ways, but there are some small subtle differences that will change the sort of like the repertoire of the peptides bound. So you might find that some people respond less well to a particular vaccine, or maybe slightly more susceptible to a particular disease. And so you can see sort of lots of them are quite similar, but then there are others like this AO220 motif, which has a really interesting requirement for a positive charge at the first position. So this K and the R that you can see. And interestingly, there's this AO252 one down at the bottom here, which is one that the peptide binding predictors don't predict particularly well. And it's really quite subtly, but strongly different from lots of the other AO2 motifs. If we look at the HLA-B peptide, sorry for the door slam in the background there, we can see that sort of for B15, which is one sort of quite interesting subgroup, it has quite a lot of different motifs. And this is a really good example of how a small number or a reasonably small number of polymorphisms can totally change the motifs of peptides bound to a particular allele. And if we look at clustering them, so we're not just looking at the cosine similarities, but we're clustering the motifs, we see that there are different families that form of them. And I think sort of the ones over on the side, the blue ones, are ones where there's only one allele in that cluster, and that's really because we don't know enough data to have found others in that particular family yet. It's very unlikely that there will be sort of, alleles that have a totally different binding motif to any other ones. Diving into sort of one particularly interesting sort of set of alleles, if you follow peptide binding motifs, you know that there are sort of 3 or 4 different collections of molecules that bind an arginine or a lysine, a positively charged residue in the center of the binding groove. But interestingly, apart from these 2 very closely related ones, B1401 and 1402, they all sit in very different clusters. And that's because of the other amino acids in the motif that make them quite different. What we also find when we look at the structural data is that actually these three different alleles, so 1402, 0801, and 3701, use a very different collection of residues in their P5 binding pockets. And in particular, the B0801 allele has a very different conformation for that central arginine. So B0801 is in magenta in the sort of like the molecular overview to the left, and we see that its arginine is pointing in a completely different direction to the ones bound by 3701 and the 14 alleles. And that's because it has this really rare polymorphism at position 9 of the alpha chain of the MHC molecule. And the reason why this is important is that if we don't have a really diverse collection of training data, we might miss out on the importance of a single polymorphism like this, ASP9. And Linda Kavraki's group have done some really lovely work where they're looking at the equity, the sort of like the ancestry equity of different binding predictors. A very important piece of work if we're to really think about how to generate a set of immunotherapies that works for all populations and not for just white Western populations. And one of the ways that you can start to sort of explore this lack of data which she's talking about is to look at what we do know, what data that we have already. And you'll see where I'm going in a minute. So if we look at the alleles for which we have motif data or structural data or a combination of both, we find that it's around 130, 140. You might feel that we're doing quite well, but if I put that into context of all of the alleles in the sort of like the sequence space, which is about 9,000, you realize that we're really not doing that well at all. And so to understand how big a problem this is, I've sort of started to look at the distance of alleles from representative data in different sets, either the NetMHCPan predictor sets or motifs or structures. And the way I've been looking at this is by looking at the relationship between the allele and the distance between them. And that distance is the Hamming distance between the NetMHCPan pseudo sequence, those residues in the binding cleft that look at the specificity between allele A and allele B. And when we look at that for the NetMHCPan data set, we find that actually that sort of diagram I showed you with the circles isn't quite as bad as we first feared, because there's sort of just under half of the alleles have a distance of 0. And so actually the training data for those alleles will cover them particularly well. However, as we move a bit further out, we find that particularly for HLA-B, you can see that there are some alleles, and quite a few actually, that are sort of 7, 8, 9, and even 10 amino acids different from this pseudo sequence of MHC residues involved in peptide binding. And considering that entire sequence is only 34 amino acids long, we can say that, you know, they're a third away almost, and so they're likely to be quite different. We do have some slightly better data in that we've got data from the MHC motif atlas, which is slightly more diverse, and there's sort of less alleles at that huge distance, but it still shows that there's information needed to make some predictors better. I should apologise for noises in the background. It's half term in the UK at the moment, so just imagine it's the middle of the pandemic and there are loads of people in the room with me.
Yeah, Chris, it's not that bad. Don't worry about that.
That's good. Good to know. So to give you a tiny bit of sort of work in progress and a bit of a sneak preview of where this information might be shown, I'm working on a resource at the moment looking at alleles and polymorphisms and motifs that will be an adjunct to the structural data, and we'll put it into a structural context. And in this resource, there'll be a page for each allele and a page for each sort of locus and allele group, along with information around the polymorphisms and whether they're a conservative change or a non-conservative change, and how often that that amino acid is found within that particular locus in the sort of like the IPD data set. And this will allow you to sort of navigate through this sort of motif and allele space a bit more efficiently than you currently can, because at the moment the data is in lots of different places, and will give you a bit more of an idea about, you know, if you have an allele group or an allele in of particular interest, it'll help you contextualize that in terms of the known information, the relationship between motifs and polymorphisms. And so this will sort of hopefully help people to answer questions that I often get asked, which is, I'm interested in this allele, what can you tell me about it? How much data do we have about it? How reliable is that data? And what's the effect of this particular polymorphism? So hopefully, you know, again, when that new version of the site goes live in a month or so, you'll be able to do a lot of the things that I'm often asked to do. So we've talked a bit about the MHC component of that barcode, but what about the peptide element? And they're very closely related, and hopefully you now understand that the peptide is and the repertoire of peptides is dictated by the MHC molecule. So if we look at peptides bound to class 1 and class 2, there are some real differences in how the peptides are bound. I'm not really going to focus too much on class 2, but to briefly talk about it, the peptides are held in this extended polyproline conformation, with the backbone of the peptide quite tightly bound by the MHC molecule itself, with sort of specificity pockets in the cleft at 4 positions. And the variability in class 2 peptides in terms of length really occurs by the peptide sort of dangling out of the ends of the binding cleft in these so-called flanking regions, some of which can contact the MHC molecule, some of which can contact the T cell receptor. So, you know, they can be important in the recognition of that peptide by a T cell receptor. For class 1, which as a structural biologist is a tiny bit more interesting for me, the peptides are held by the ends, but then the middle of the peptide is really can be in a large number of different conformations. And if you look at it initially, you might think, you know, that's an awful lot of noise. And just to focus on them in isolation rather than just showing them next to class 2, we can see that the N and the C termini are pretty well held. And we've known this since some of the really early structures from, you know, people like Don Wiley and Ian Wilson. But the middle of the peptide seems to be, you know, almost infinitely variable in terms of how it's organized and its relationship with the MHC molecule. If we want to make sense of this noise, previously people have looked at things like phi-psi angles, so the torsion angles in the peptide backbone, and you can cluster a bit with those, but you tend to get really quite broad clusters. So we've been playing with these things called voxels. And so voxels are volumetric pixels. They're little sort of squares. And we can build a voxel grid in the binding cleft of the class 1 molecule. So in step A, we find the centroid of a class 1 molecule to which all of the other peptides are sort of aligned to that one. We can then build some XYZ planes in it and build a bounding box. And then in position D, you can see that we can find the voxel coordinate for, in this case, every C alpha atom within the peptide. And if we look at those C alpha atoms, we can see that actually positions 1, 2, and 3 are fairly conserved in their location in the binding groove, and position 9 is, but it's sort of very variable in position 5 or 6. And if we look at graphs of voxel occupancy, it's even clearer that positions 1, 2, and 3 are quite conserved in their location and thus their voxel usage. Same with position 9, to a certain extent position 8 as well, but the middle of the peptide is quite different. So the nice thing is that this voxel representation recapitulates things that we know about the structures. The other nice thing about voxels is that they have neighbors. So if we know the position of one atom, we can sort of see if the atoms of similar type are in a neighboring voxel. And again, it's even clearer that positions 1, 2, and 3, you have almost 100% occupancy of sort of like the mode voxel and its neighbors. Same thing with position 9, and then this variability in the middle. And if we do some DB scan clustering of the different voxel locations, what we can see is we get some really beautiful conformational families fall out. So we get some really strong conformational families for peptides that are anchored in the middle, those P5 anchors, like the arginine example I gave you earlier. You get some other ones for P6 anchors, which you see quite a lot in mouse class 1 molecules, but some human ones as well. And then you see a whole collection of different peptide shapes for molecules that have no central anchor at all. And so we have these really lovely conformational families, and we've been sort of thinking about how we use those conformational families, but also where we might find sort of similar information within other structures. And a piece of work I'll briefly talk about is a piece that I've been involved in with Hashem and a DPhil student called Benji McMaster, who's in Hashem's group, and also with Jamie Rossjohn and Charlotte Dean. And what Benji did was he created a data set of peptide MHC complexes that were available in both the APO and the hollow form with a T-cell receptor. And I'm not going to dive too much into this work, but in terms of this presentation, the really interesting thing around it is if we look at what moves when the complex formation occurs, actually we find that the MHC molecule, so if we look at this plot on the left, the MHC molecule in both in a TCR contact position and a non-TCR contact position moves very little, but the peptide moves significantly more on complexation. What's more interesting is that actually these movements that you see in the peptide relate to the anchoring strategy. So in the complexes that I showed, in the conformations I showed you earlier, you had distinct conformational clusters for peptides that were anchored at either end, at P2 and P9, but weren't anchored in the middle. And when you look at how these peptides move when they contact the T-cell receptor, you get quite a massive change in that central, very flexible 4, 5, 6, 7 area that we talked about earlier. In contrast, in the right-hand side of this plot, when the peptide is anchored in the central position, in the P5 position, we see movement at either side of that anchor. So we see quite a different pattern of movement there. What was also really important in sort of the work that Benji looked at was that we found that there were sort of distinct contact zones for the different alpha and beta chains, and that the peptide is contacted by both, which has some real implications when we're looking at antigen specificity predictors, which previously have looked at quite blunt instruments like just the beta chain CDR3 regions. Coming back a bit more to the peptide binding cleft to the MHC molecule, another way you can use these voxels is you can look at the water molecules that are bound in the binding groove. So the one thing you notice as soon as you start looking at these molecules is that the binding grooves are totally full of water. And if we look from the side, a lot of the water molecules are above the peptide, so they're in sort of the T-cell contacting area, but a large number sit underneath it. And actually, if we sort of delve into water molecules that are found in greater numbers of structures, like ones that are found in more than 2% of the structures, we find that there's some really quite conserved locations for water molecules that are involved in holding the termini of the peptide. But there are other water molecules that we want to explore a bit more about how they relate to different alleles or different peptides. And we might want at some point to use this data for machine learning of water molecule locations. And the reason why we want to do that is that we can maybe reduce the equilibration time for molecular dynamics simulations. We can find locations where metals or drugs might bind within the binding cleft and displace these water molecules. But we also want to improve the electrostatics because in predictors such as AlphaFold, there's really no measure of electrostatic at the moment. You can put it in as a loss function, but it's not implicitly known by AlphaFold. So we think that understanding water molecules and where they bind and how they bind is important to understand peptide flexibility and lots of other things as well. briefly moving on to AlphaFold, I thought I'd give a bit of an example of what works, what doesn't, and why that might be. And if you look at all of the sort of like the sort of like the general purpose scientific press, you might feel that AlphaFold has solved everything. And it's solved a lot of things, and it's a massive leap forward, and I'm very grateful for it. But actually, it hasn't solved everything in the immunological context. And if you'd like to read a bit more about this, there's a Perspectives article that I was fortunate to be a co-author on with Benji, Graham Ogg, Charlotte Dean, and Hashem. So I encourage you to go and read that. And we need AlphaFold because we've got so little information structurally compared to the wealth of protein alleles. We're going to need structure predictions to predict things at repertoire scale, but we're not at that position quite yet. And actually, if we're to give AlphaFold a sort of end of term scorecard, we can say that it's really great at just the MHC molecule structure, and that allele resource I was mentioning earlier has a prediction for every single allele in there. And I'm now working on the version of that for all species, not just for humans. So there'll be also over 20,000 predictions eventually. It's improving for the MHC plus peptide, which will be the focus of the last bit of this talk, but it really needs to try harder when you include the TCR. So briefly talking about AlphaFold, there are two important metrics when you're trying to benchmark it. And the first is around how accurate it is compared to another structure, so the ground truth structure. And for that, we use this root mean squared deviation metric, which tells us how close two structures are in 3D space. But the other important metric is this confidence for AlphaFold. So how confident does it feel about its prediction at that precise location? And that's this PLDDT measure. And if you'd like to learn more about AlphaFold, there's a really great online training course that you can take on the EBI website, which I encourage you to look at. And if we're to look at how AlphaFold is improving over time, this was the first version of Multimer, so the first version that we could predict complexes with. And for the bound peptides on an MHC molecule, it did really well for a very, very small number of cases, but it did pretty terribly for others. And if we look at how it's improved over time with those same peptides, as we move into the middle panel, which is the slightly better version of the multiple weights, it's really improving, and then it's getting much better for AlphaFold 3. And if we look at sort of ones that it did really, really bad with, again, we're seeing a massive improvement. And there are some where there's still some room for improvement. But the great thing is that AlphaFold now gives you these sort of per atom confidence scores. So if you see something that is sort of is a very different color, when you visualize them by color, you realize that it's less confident about that particular part of the complex. Looking at sort of how it's improved in terms of sort of putting these RMSD values into sort of different quality bins, we can see that the majority of structures that are predicted by AlphaFold in terms of quality are in this sort of excellent bin now. And if we look at the violin plots, you can really see the change in the quality of the peptide binding prediction across the different versions of AlphaFold as we move towards AlphaFold 3. The one thing I should say with AlphaFold 3 is it actually has the peptide included within its training data set. AlphaFold didn't have that. And so these ones in this comparison are all peptides which are at a reasonable homology distance, sort of 4 changes within the peptide, so that we're trying to keep well away from the training data set. Just looking at the ones it does really well on, you can see that there's really high agreement between the predictions and the ground truth structures. And the ones where it doesn't do quite so well on, we can sort of start to understand. They're either ones like the sort of the second one in from the left on the top row, where there are quite a lot of glycines in the middle of the peptide, so it's really high in terms of flexibility. And then if we go for the third one in from the left on the bottom row, there's a lot of arginines in that peptide and lots of sort of charged residues. And as I said, AlphaFold doesn't really understand electrostatics yet. But one of the things to note from this is that we're beginning to run out of structures to benchmark with. So in the training data set, there are sort of 412 structures before the date cutoff. And in the bits after the date cutoff, we've really only got 37 structures we can benchmark with. And in total, we're doing okay in terms of training data compared to other molecule types. So there are over 1,000 unique peptide MHC class 1 structures in the PDB, which has got just over 200,000 structures. So MHC is pretty well represented in there, but actually it's a bit of a rounding error when we compare it to the size of the repertoire. And again, there's not much diversity in there. So the diversity is in terms of peptides is really, really low. You have lots of peptides related to each other in there. So we need to really focus on this diverse aspect of it. But if we're to look back at this scorecard, we can say it's doing really well for the MHC molecule, it's doing better for the peptide, and it must try harder for the TCR. And why is that? Well, I believe it's partly because of the amount of co-evolutionary data there is in there, and I'll come to why that's important in a minute. So for the MHC molecules, we've got lots of structures from many species, and the co-evolution is really well represented. For the peptide, that co-evolution isn't represented at all now, and it's definitely not for the complex for the TCR. And to illustrate what this co-evolution is, this is a diagram of how AlphaFold works, and the co-evolutionary information comes from these things called multiple sequence alignments. So the box that says MSA there, and that's basically how a molecule has changed over evolutionary time. And there's lots of signals in there about which parts of the molecule are close to each other. But for peptide MHC complexes and for peptide plus MHC plus T cell receptor, the co-evolution doesn't happen over evolutionary time. It happens in the sort of like the interaction between a peptide repertoire and an MHC molecule, or the antigen T cell receptor relationship. And this co-evolutionary information isn't represented within the sequence alignments. So I just thought I'd finish today by sort of showing you a bit of experimental work where I've been trying to teach AlphaFold a bit about this sort of co-evolutionary information. And I started off by giving it a very small amount of co-evolutionary information to see if I could break it initially. And obviously the predictions that you see on the left are really quite poor compared to the MHC molecule. It's still got beta sheets and helices, that's great, but the structure really isn't there. And if we look at the plot on the right, we'll see that there's really poor PLDDT. If you give it the membrane proximal immunoglobulin domains, but still a very small amount of co-evolutionary information, it does slightly better, but it does really well on those immunoglobulin domains. And what that tells you is that AlphaFold implicitly knows about an immunoglobulin fold. If we start pulling apart the MSAs you get from the Colab fold program, which is a sort of an easy to use version of AlphaFold, and you try and deconstruct it and make it simpler, you see that you can start to recover some of the co-evolutionary information for the antigen binding domains. And if you then give it a bit more information, so on the right here, I'm giving it information on one sequence for every locus for every species in the IPD data set, we can recover pretty much the PLDDT graph, and it now feels really confident with values sort of between about 80 and 100 for PLDDT for most positions. What's really interesting is if you give it all the information in the immune polymorphism database, so if you give it 24,000 sequences, so one for every allele in that collection, it starts to become less confident. And so you've given it too much information and it now can't find the co-evolutionary signals. And so what I'm starting to do now is to sort of do different homology level cutoffs to see if I can find a sweet spot between too much information and too little information. But if you look at the quality of the predictions that come out from this little experiment, you'll see that actually the peptide is really well structured. The MHC molecule, as we expect, is really well structured. And actually, the side chains of the binding cleft residues are well structured as well. But one caveat around AlphaFold, and it comes back to this sort of observation from Benji about the sort of like the role of anchor residues and the mobility of the peptide on complexation, is you only really ever get one complex out from AlphaFold. And so we're going to have to work out how we would give it information on the sort of like the allosteric changes that happen on complexation. And then finally, to sort of say where next with the AlphaFold work, we can potentially start to look at peptide binding affinity now that we're getting these PLDDT values for every atom in the complex. Along with Graham-Ogg's group, we're looking at lipids bound to CD1 and how we can use some of these machine learning predictors for that. Again, it's early days, but the jury's slightly out as to how well they do. We can look at peptide MHC, TCR, just to keep an eye on how it's doing and how we can improve it. And then for class 2, it seems to work quite well for some structures, but it gets very confused if you give it too much in the information about the flanking regions. And so again, there's places to tune it. So finally, a few quick conclusions. We've got a lot of data, but it needs a lot of curation and linking, and a large part of my job is doing that, because for machine learning we need clean data in a very amenable format. We've got a lot of data, but we don't have enough. It's very easy for me as a bioinformatician to say that, whereas it's much harder for people who are doing structure determination. But we need more data, and we certainly need to fix the diversity. We probably need to move beyond sequence-only methods. I think sort of if we look at lots of the things you can do with sequence, like peptide binding predictors, they're starting to sort of hit a limit of what they can do, especially around generalization. So we'll need to think about other data sets we can add in, such as structural data. We really, really need to decolonize the data. Until we have true generalizability, we need to make sure that we can predict not just white Western immune responses. Otherwise, we won't have equitable health across the world. And then finally, I think structural data can provide some of the context that seems to be missing to these AI methods and may well help with efficiency. And there may be other data modalities we can use, and we really need to think about how we can use some structural data. And finally, AlphaFold is great. It's very exciting. We're not quite there where we can trust it for everything to do with immune complexes, and there's still far more work needs to be done to improve it, to tune it, and we'll need a lot more structure determination. I briefly wanted to mention the hackathon that Hashem trailed earlier. It would be lovely if any of this has inspired you to come and work on machine learning on MHC molecules. And then finally, sort of thanks to all the people involved in this work, both at the EBI, University of Oxford. I'd like to also thank John Jumper, who, you know, as a very busy person, even before he had the Nobel Prize, would always answer emails, which was really amazing. And I'd like to thank Viridian, who provided some compute resources for the work on tuning AlphaFold. So I'll stop sharing now and open for any questions.
Thank you so much, Chris. Lovely, really. You know, one thing I really love about your seminars and discussion with you, and I'm really always impressed, is the depth of your work, both at immunology side and also in technology side, you know, all those AI, and you are truly a good example of you know, type of work to sit at this intersection. So thank you so much for that.
Thank you,
Yeah, so please